Bueno, pues nada, asumimos que ya estamos online, ¿no?
Yo creo
Bueno,
que sí.
pues muy bien. Buenas tardes a todos y bienvenidos a este nuevo seminario del y en este caso, pues también del área de oncología. Y I'm going to switch to English to introduce Pilar. It's for me a great pleasure to have here Pilar. She, in addition to a good colleague, she's also a friend of mine, and we met a long time ago, and I am glad that now our research interests also come together, especially with the brain tumors, and you will see that she has a very nice story to tell us. She has always been like a neuro person. I mean, doing her PhD in always in the nervous systems and trying to understand the molecular mechanism between the nervous system, the neural stem cells, and then she has been moving more recently to the neural brain tumors. She did her first postdoctoral stage in the Skirball Institute from the New York University. And then after a second postdoc in the Geneva University, she came into the Spanish system through a Ramón y Cajal contract in the University of Valencia, also in the group with Isabel Fariñas, where we had the opportunity to meet in a I think it was a muerte neuronal and neurodegeneration meeting.
I don't remember.
No, where we met her, when I met her in Valencia, and when it was one of these moments in she changed my life. No, so she recommended me, introduced me my postdoctoral mentor in NYU. So and this was a big change in my research career. But after this after this Ramón y Cajal period in Valencia, so she became an independent scientist and become the founder of the neuro-oncology unit at the Instituto de Salud Carlos III, where she has been developing her research. If you hadn't had the opportunity to look at her publication record, you will see that she has always been a top scientist with very nice work that we all enjoy to read. So please, Pilar, the stage is yours. So, and I forgot to mention that everybody that is interested in asking questions, so please go to preguntas on the right part of your menu, and there you can write your questions. And after the talk finish, I will read it to her, and we can have a discussion. Okay, thank you. So, Pilar, whenever you want.
Thank you, Miguel. Thanks for the invitation. Whoops. Can you see the screen? I can see thousands of screens. Miguel, can you see the screen well? Okay, I hope you can see me, because I don't see you anymore. So thank you for the invitation here to talk at the Vall. You actually invited me exactly 1 year ago. We had to cancel because of this terrible year. And I know it's been a terrible month for many things in life for all of us, but actually for our lab, it's been a very productive and a very successful year. So I must think that maybe we have to spend more time thinking and not so much time on the bench, maybe. During the last year, we have been working on gliomas actually for a long time, all the time in collaboration, at least for the last 11, 12 years, in collaboration with Hospital Universitario 12 de Octubre. We have been working on gliomas. And in the last years, our focus has moved more into understanding the biology of the microenvironment of the tumors. The special microenvironment of the brain is conditioning the growth of these very aggressive tumors. These are my disclosures. And just to start with a very general thing, we all know that the incidence of cancer is increasing, but at least in the developed countries, the good thing is the mortality is decreasing, but this mortality is not decreasing for all the cancer at the same rate. And particularly for brain tumors, there's been not much advances in the last 10, 20 years. Brain tumors is also a very heterogeneous group of cancers, depending on the cells of origin of these tumors. And we are particularly interested in gliomas, those of glial features, which are one of the most common among these primary brain tumors. Among gliomas, 50% of them are high-grade gliomas. These tumors have a very, very aggressive behavior. Fortunately, they are not very common. They are a little bit more common in adults than in children. But these tumors are actually not curable because in 100% of the cases, they recur and they end up with the life of the patient. And I want to look to this graph. It's a little bit old, but I think it's clear that even if the survival of the younger patient is a little bit better, this is survival at 5 years after diagnosis, this survival has not increased, not for younger patients, not for adults, in the last decades. And actually, there is no good treatment for gliomas, for high-grade gliomas. Right now, the current treatment is what we call the Stuck protocol, which is an aggressive surgery followed by radiotherapy combined with temozolomide. Temozolomide is an alkylated agent that is going to kill the cells in division, and it has improved a little bit the survival, the average survival of the patients. But you see, not too much. But the thing, this drug was approved around 2000, year 2000. And from that time to now, only this other, is not a drug, it's a device, this TTF, this tumor treating field, is a helmet that induces electromagnetic waves in the brain, so it stops the cells in division. And it's the only thing that also has proven some benefit for the patient. And the bad thing with this helmet is actually not many patients accept to take it, and it's very expensive. So it's actually, at least in Hospital 12 de Octubre, almost all the patients are being still treated with the Stuck protocol. And then, of course, when the tumors recur, they enter into clinical trials or they receive the bevacizumab, the anti-VEGF therapy. But there is actually no good therapy for these tumors. Although there has not been advances in treatment, what is very clear is the classification of the tumors has improved a lot in the last decades. And the WHO classification has included now many molecular parameters, and probably the most important one is that one of the finding of these mutations in IDH1 genes. These genes, the isocitrate dehydrogenase enzymes, these are enzymes that participate in the metabolism. And when they are mutated, actually, there is a conversion from alpha-ketoglutarate into 2-hydroxyglutarate that accumulates in the cells. And this metabolite is going to end up activating the hypermethylation of the DNA and histones. And this is supposed to be related to the tumorigenic effect of these mutations, not only in gliomas, but also in leukemias or myelomas. And this is associated in gliomas with this G-SYM phenotype. It's a hypermethylated phenotype that is very also easily detected with DNA methylation patterns. So the good thing about these mutations is the most common one in 80%, 90% of the cases is this mutation, and there is an antibody that works very nicely in immunohistochemistry. So almost all the hospitals are now doing these classifications because it distinguishes two different entities, very different entities. It distinguishes the IDH mutant gliomas from the wild type. And these IDH mutant gliomas are less aggressive. And it includes tumors with astrocytic features and all those with oligodendrocytic features. And then you have several degrees depending on the percentage of atypia, the proliferation of the tumor cells, and also the appearance of things like necrosis and vascular proliferation here, which increase the grades. Here you have a grade 4 astrocytoma. It appears normally associated with mutations in P16, P19 genes. And the curious thing is that these tumors histologically is very similar to this other one. It's what is now called the glioblastoma. It's an IDH wild type grade 4 glioma. And histologically, they're actually very, very similar. The pathologists are not going to be able to distinguish it. But the prognosis and the behavior of these tumors is very different. So the ADH wild type are much more aggressive, even though these ones are also aggressive tumors. So nowadays, it's well accepted that this IDH distinguishes two different diseases, at least for gliomas. Also, gliomas were one of the first tumors to be studied by the TCGA, to be sequenced. And also the several transcriptomic analyses have been done, and it's more or less accepted that there is at least three transcriptomic groups: the proneural ones, the classical, and the mesenchymal. The neural is now that it is now accepted that it's probably contamination with normal tissue. So proneural tumors express a lot of these genes, SOLIC2, DDL3, are more similar to, well, we used to say the progenitors, the normal neural progenitors. In the mesenchymal tumors, there is a lot of expression of things related to extracellular matrix, of IKL40. And in the classical tumors, there is a lot of expression of EGF receptor, nesting, a lot of proliferation. They're also sometimes called proliferative tumors. If we look back into this classification based on IDH mutations, we can see that proneurals include all these gliomas or glioblastomas, with IDH mutations, although a few of them don't have these IDH mutations and are very aggressive tumors as well. Among IDH wild-type gliomas, they concentrate these two transcriptomic groups, the classical and the mesenchymal, which are associated with a slightly different accumulation of mutations. For example, EGF receptor mutations accumulate in the classical ones. By contrast, NF1 mutations and things related to extracellular matrix accumulate in the mesenchymal tumors. So as I told you, our lab has been interested in studying the glioblastoma in microenvironment during the last years. And one of the things that drives our attention a lot is the vascularity of the gliomas. Because glioblastomas are considered one of the most highly vascularized tumors of all. You can see here this 3D reconstruction. This tumor here is highly innervated with vessels. With the endothelial cells are proliferating and they form these glomeruli structures. You can see it here on the right side. In a 3D reconstruction, you can see these vessels. The funny thing is, many times, even though they have so many vessels, these vessels are not highly functional. Even more, many times there is thrombotic events that saturate these vessels and they collapse, and there is these areas of necrosis that appear. And this is a typical feature of a glioblastoma, which is called pseudopalisade. Is these areas of tumor cells that are migrating and proliferated around this area of necrosis, of course, an area with highly density of hypoxia signals. Other times, there is highly massive areas of necrosis, what they call extensive necrosis. So these tumors have these two features very clear: highly vascularized with this glomeruli and enriched in these necrotic areas. And another important thing to understand the vascularity of gliomas is the thing of the blood-brain barrier. This is from a review we wrote recently. It's trying to describe what is the Neurovascular Unit. So in the brain, the vessels, the small arteria and venous, are covered by endothelial cells, of course. And these endothelial cells are covered by pericytes. And the ratio of pericytes to endothelial cells in the brain is much higher than in any other tissue. So there is a lot of pericytes. And these pericytes, you're going to hear me talking about them a lot, is funny cells. They are mesenchymal in origin. They are very plastic, and sometimes they are confused with fibroblasts and with other mesenchymal cells. And they're supposedly to have a strong function here in the neurovascular unit, regulating all this structure and secreting extracellular matrix and signals to regulate. Also, in this neurovascular unit, you have the feet of the glial cells surrounding the vessels. And of course, all this structure, what is regulating is the function of the neurons, which are also connected to this neurovascular unit. And microglia, macrophages in the brain are also interacting with these structures. So what happens in the gliomas? In a high-grade glioma, particularly in glioblastoma, one of the distinctions from other lower-grade tumors is that this blood-brain barrier gets disrupted. So, for example, you have here a three-dimensional staining. You have here the alpha-SMA staining of the pericytes. In a tumor, there is a lot of pericytes that get detached from the vessels. Also, the same connections between endothelial cells are lost. So the blood-brain barrier gets disrupted, and that's the way to distinguish these tumors through the MRI images. Here on the top, you have a typical image of a grade 3 glioma. You can see in the T2 flare the invasive cells around this area. But then there is no extravasation of gadolinium, there is no contrast enhancement. But this is the typical image of a glioblastoma with this necrotic area in the center, surrounded by a vascularized rim, and around it, this called area of edema, which is a mix of vascular edema and also invasion of cells. And actually, this is a funny area that we are now interested in looking at. So the mechanisms of vascularization in glioma are very well described, and they're supposed to be like a sequential. So it's supposed to happen at the beginning of the growth of the tumor, that the tumors grow around the persistent vessels in a process called vascular cooption. And then the tumor cells start secreting angiogenic molecules, particularly VEGF or FEF, and forming this glomeruli and a lot of little vessels. They're also recruiting progenitors from the bone marrow in this process of vasculogenesis. And then the glioblastoma cells are also highly plastic, and they are able to form these tubular structures that mimic the vessel. It's called vascular mimicry for that. And even they can transdifferentiate into endothelial cells, and they actually start expressing some markers of endothelial cells. More recently, the group of Vall and Jeremy Rich have described that the glioma cells have a strong capacity to differentiate also into pericytes. You can see here in the models how these tumor cells in GFP are surrounding the vessels and expressing markers of pericytes, and how these pericytes have mutations typical of the glioma cells. And this is not something sporadic, as they have been able to measure that even 70% of the pericytes in some of the tumors they have looked are actually neoplastic cells. So what they believe he's doing is these pericytes from the tumors are reinforcing the vessels. So in a way, they're making this vessel more functional, but in a way also they are reinforcing the blood-brain barrier. So what they propose in this other paper, more recent from 2017, is that they could target this pericyte with ibrutinib, which is an inhibitor of VMX, and they show that VMX is controlling these tumor-derived pericytes. So with this ibrutinib, they propose that they could open the blood-brain barrier, facilitating the entrance of other drugs. And actually, in 2018, they started this clinical trial combining ibrutinib with temozolomide, and it's still going on, so I don't have still the results, but I'm really interested in knowing. And this is not just particularly for gliomas, because there's been reports of this transition into pericytes. It's also called epithelial to mesenchymal transition in other cancers. And it's been shown in renal cell carcinoma or also in breast cancer. You can see here that these tumor cells lining the vessels in those tumors with some mesenchymal features, and not in other, like this well-known model of breast cancer. So you see that these GFP tumor cells are lining the vessels, and they're expressing pericytes markers, and they can follow them also with some reporters. Unlike in gliomas, where they propose that these tumor cells, by lining the vessels, they are promoting more vasculature, they are promoting the vessels are more functional, so they are promoting the proliferation of the tumors. Another difference of gliomas from other tumors is the unique immune microenvironment, and this is because of the unique immune microenvironment of the brain. You know, the same blood-brain barrier that prevents the entrance of other drugs into the brain is also preventing the entrance of immune cells in the brain. And you can see here in this pan-cancer analysis, actually, it is very clear that both glioblastoma and low-grade gliomas are very highly different from other cancers. Basically, they are either lymphocyte depleted or immunologically quiet. Basically, low-grade gliomas have very little immune cells inside, whereas in glioblastoma, there is little lymphoid cells in the tumors, and the lymphoid cells are, of course, exhausted. But there is a lot of macrophages and myeloid cells, myeloid-derived suppressor cells, all with pro-tumor features. So this is also very strongly, it makes very difficult to treat these tumors with immune therapies, which actually hasn't worked so far. So what we have been doing in the last 2-3 years is trying to connect what happens, how the genetic background of the tumor cells, how the tumor cells are controlling the tumor microenvironments that surrounds them. And we have been focusing on the vascular, but also we have done some studies in the immune features. Before I started, I would like to say some of the tools that we use commonly in the lab. So we normally work with primary cell lines, and we grow them in the absence of serum, as we call them neurospheres. We grow this form of beans of balls of cells. And we derive these patient-derived xenografts in nude mice. And also we are going to be mentioning it a lot, some tools for blood-brain barrier analysis. I'm going to be showing a lot of markers for endothelial cells like endomucin, CD31, and also markers for pericytes like alpha-SMA, and particularly CD248, which is a molecule that is specific for tumor-derived pericytes, and it seems to be controlling the expression of the other markers. And also we have used some markers specific for the tumor cells. And I would like to mention also 2 mouse glioma models that we derive in our lab. And we derive them from the subventricular zone. We extract the subventricular zone from mice that were P16, P19 cow. And we derive these progenitor cells, and we express them with lentivirus, the EGF receptor, either wild type or the B3, to generate these 2 cell lines. And why these 2? Because EGF receptor is one of the biggest targets in glioma because it's overexpressed in more than 50% of the glioblastoma. In most of these cases, it's amplified. And this amplification leaves with a lot of mutations, either point mutations or deletions. It's important to point out, for those of you that work in other cancers, that mutations in EGF receptor in glioblastoma are very different from, for example, the lung cancer mutations, which accumulate in the tyrosine kinase domain. In glioblastoma, these mutations accumulate particularly in the extracellular domain. And the most common one is the EGF receptor variant 3, V3, which has a strong deletion in the extracellular domain, which makes them, the receptor doesn't need the ligand to be activated. Of course, being one of the most important targets in glioblastoma, there's been a lot of clinical trials trying to inhibit this molecule, this receptor. And we have participated at least in one of them in the test from Pfizer, testing dacomitinib. And I have to say it's been a little bit disappointing because most of these trials hasn't given any good results, and only very few patients have benefit from that. There's been also some attempts to target specifically the EGF receptor B3, which is expressed more or less like in 30-40% of the glioblastomas with specific antibodies, and again, no good results. So for us, it was important to have these two models because they really represent the disease. This P16P90 cow deletion is very common also in gliomas, together with these amplifications on the EGF receptor. And as you can see here in our model, the B3 is more tumorigenic than the wild-type EGF receptor, like the same way it happens also in the human tumors. So going back to what I wanted to tell you is two papers that we have recently published, and they are very well connected. And the first question we try to answer is, okay, so IDH mutations give rise to tumor. They have the tumorigenic action of IDH mutations have been well explained, but why these tumors are so different from the others? Why are less aggressive? So this is something that has been performed mostly by Riccardo Garghini and Berta Segura, and we published last year. So we were looking at one molecule, it's called tau. Tau, the gene is called MAPT, and I'm sure all of you know tau, especially if you work in neurons, because it has a plethora of functions in the neurons. It's a microtubule binding protein that stabilizes the microtubules. It participates in the synaptic transmission and the movement of vesicles. But of course, all of you know that mutations in tau or other mutations in other protein that becomes the protein, the tau is phosphorylated and accumulates in oligomers and tangles. And this is associated with several tauopathies and is associated with Parkinson and Alzheimer. But when Ricardo came to the lab, he was coming from the lab of, from the CBN. They were experts in Jesus Avila, from the lab of Jesus Avila, and they're experts in tau in neurodegeneration. So he was looking at the expression of some of these neurodegenerative genes in gliomas. And he came to observe that most of these genes were more expressed in the gliomas grade 2 compared to gliomas grade 3 or grade 4. It's like there is an inverse association with the progression of the disease. And also, for example, there is association with a good prognosis in most of the gliomas. And the strongest association was observed with MAPT, with tau. So we started looking at several cohorts. Of course, in the TCGA, you also see that there is a decrease in tau expression as long as the tumor progressed from grade 2 to grade 4. We looked in the tumors, and the tau protein is expressed actually in the tumor cells and not just in the neurons. You can see here, this is a typical staining of tau in a normal brain, and this is a neuron here. And this is the expression in the tumor cells, and it's more expressed in the lower grade tumors compared to glioblastoma. Of course, it's associated with a good prognosis. We looked also in our models, in our PDX, and we have, I told you, we have been working with primary cell lines for a long time, and we have a battery of cell lines. And you can see here they have varying degrees of expression of tau. So in all the lines that express high levels of tau, these are less aggressive. And all the lines that have a high expression of tau, the tumors are very aggressive. We also were able to look at cohort of samples, small cohort, but I think it's very interesting. It's tumors that were from low-grade tumors, and they recur, and they actually recur without progression, just stay with the same degree, or they progress into glioblastoma. So all the time the tumors became more aggressive, developing into glioblastoma, there was a decrease in the expression of tau. Whereas if the tumor stays stable in the same degree, tau levels are the same. So we really believe that tau is like a break point for the gliomas to progress into a more aggressive tumor. Through the bioinformatic analysis, we noticed also that tau high expression was accumulated in tumors with IDH mutations. So we looked in vitro, and for example, the 2-hydroxyglutaric, which is the metabolite that is induced by IDH mutations, induced the expression of tau. It looks like it stabilized the protein. But also in vivo, we were able to express IDH mutations in a model, in a mouse model. It clearly induced the expression of tau, and of course, it makes the tumors less aggressive. I'm not going to go much into detail, But just to tell you that we know now that there is an epigenetic regulation of the MAP2, the MAPT, the tau promoter. And I know it sounds counterintuitive, because normally methylation is associated with downregulation of expression. But there is particularly some genes, and this paper was published in Nature in 2016, and regarding the regulation of PDGF receptor also by IDH mutations, and they found that there is this island where CTCF is bind. So when this island is methylated, it allows the enhancer to bind to the promoter, and allows expression of the gene. So we found two regions in the promoter of MAPT that were hypermethylated in the presence of mutation, and then the binding to CTCF is lost. So what is doing tau? What happens when tau is overexpressed? What we observe is that there is a downregulation of EGF receptor signaling, but it only happens in the absence of mutations. You see here that if the tumor has B3, but also point mutation, it happens the same. Tau doesn't have any effect in the aggressivity of the tumors, doesn't have any effect on EGF receptor signaling. And it seems to be mediated through changes in the stability of the tubulin, in the amount of acetylated tubulin that controls the turnover of EGF receptor. So in the presence of tau, the tubulin is highly acetylated and EGF receptor is degraded. We also knew from the in silico data that there was an inverse correlation between MAPT expression and the mesenchymal markers, and a positive correlation with the proneural markers. So in the tumors that express tau, we observed that there was a decrease in the phosphorylation of NF-kappa B, or in the stability of YAP-TAS, which are two transcription factors associated with the mesenchymal features of gliomas. And this signaling does not change in the presence of mutations. Parallel to this, you see an increase in proneural markers. So in the presence of tau, there's been a shift from mesenchymal features to proneural features. And there is a strong effect in angiogenesis. We just noticed when we were extracting the brains from the mice that tumors without tau, many times they have these big hemorrhages. They do not appear when tau is overexpressed. And there is a big change in the vascularity, with a decrease in the number of big vessels, of dilated vessels, a decrease in VGF, and also a strong decrease in the stabilization, for example, here measured by stabilization of IgG into the tumor. So this is like the cartoon of this paper. We know that the balance between the amount of IDH wild type and IDH mutant is what controls tau expression, tau stability. So in the presence of mutation, there is high expression of tau, which induce microtubular stability, blocks EGF receptor, and blocks these signals that drives mesenchymal transition of the tumors, and the tumors have a more normalized vasculature. When IDH mutation disappears, or there is a lot of expression of IDH wild type, there is very low expression of tau, and then this signal, EGF receptor, NF-kappa B, TAS, all of them increase the mesenchymal signals associated with angiogenesis and with disruption of the blood-brain barrier, and then the tumors become very, very aggressive. At the same time, and this was a little bit, it was studied a little bit later than these results, we observed that the MAPT expression was inversely correlated with a lot of features, a lot of signatures of immune cells. This is a work that we did, was led by Dr. Sepúlveda at Hospital 12 de Octubre. So they were doing this flow cytometry quantification of immune cells from the tumors of the patients. And we noticed that there is an inverse correlation between the expression of tau, which is highly accumulated in IDH mutant tumors, and the presence of immune cells in the tumors. This paper, of course, has much more data, but just as an example, we were able to express tau in GL261, which is a common mouse glioma cell model. So when you express tau, the tumors grow slowly, and there are changes both in the vasculature. You see less vessels. You see less dilated vessels in the tumors, but it's also less CD3 cells in the tumors, less immune cells entering the tumors, and less expression of immune cell markers. So it looks like tau is controlling both things, the vascular features and also the immune feature, and it's making the tumor with a more normal vasculature, but also with less infiltration of immune cells. Both things, we believe, they contribute to the less aggressivity. And the other story, which I think is connected to this one, is also connected to the regulation of the vasculature in the tumor, is what happened in the IDH wild-type gliomas. Could it be that EGF receptor mutations is modifying this? But because we knew that B3 mutations is more aggressive, so we went to see how were these tumors. You can see here clear there are very different tumors. The B3 mutation-induced tumors with a high cellularity, a lot of BRDU incorporation, a lot of proliferation, and also a lot of these high dilated vessels in the tumors. When we design a vascular signature, we saw that it accumulates in the presence of the mutation. And something that surprised us a little bit is that we went into the MRI with these mice, and even though these B3 tumors are more aggressive, when we look through the MRI, even though you see some extravasation of gadolinium, the extravasation is much higher in the tumors without the mutations. And you can validate that seeing IgG extravasation, which is very little in B3 and is very high in the absence of mutation. And this extravasation of the IgG, this leakage of the blood-brain barrier, is associated with an increase in hypoxic signal that you can see here by accumulation of HIF1 alpha, or by the expression of a hypoxia signature that we define in the tumors. So what is happening in the B3 tumor? What is happening in the presence of EGF receptor mutation that prevents this leakage of the blood-brain barrier? So we look to see if there were happening transdifferentiation of the tumor cells into endothelial cells, and we didn't see much difference there. But we did see is an accumulation of parasite markers in the tumor cells in the presence of mutations. And this is very nicely seen here also in the PDX, because we can distinguish the host parasites from the tumor parasites with specific primers. So we see that there is accumulation of human parasite markers, but the mouse parasite markers are not accumulating. So for us, it's very clear that in the presence of the mutation, there is a stronger induction of transdifferentiation of the tumor cells into parasites, which actually increase the parasite coverage, decrease the leakage of the blood-brain barrier. So it's like it was supposed to do. These parasites are reinforcing the vessels and are making the tumors more aggressive. You can see here, if we overexpress B3 in a tumor with amplification, we make this tumor more aggressive. And if we deplete these parasites with a human-specific RNA interference for CD248, we revert this phenotype. So really, this transformation of the tumor cells into parasites are making the tumors more aggressive and are driving the tumor growth. Downstream of EGF receptor, we observe that there is an accumulation of SOX9. SOX9 is a transcription factor clearly associated with some neural progenitors. We saw that it accumulates in tumors with EGF receptor. It accumulates in protein, but also accumulates in mRNA. It looks like there is, in the presence of B3 or point mutation, there is a stabilization of SOX2 protein that doesn't happen in the absence of the mutation. And we can, if we use we study tumors with the point mutations, like this tumor here, and we interfere SOX9 expression, we can make this tumor less aggressive, and we can downregulate of these markers of human parasites in the tumor. So, and also we did more tests, and it's also promoting that the blood-brain barrier gets disrupted, and it does induce hypoxia. So it really reverses the phenotype into more like an EGF receptor amplified phenotype. So just to conclude, to make all the things together, we came out with this stratification of glioblastomas based on this vascular and also based on these mutations. So it's well known, it was well accepted that these IDH mutations have a more normalized vasculature, and we believe this is mediated by tau. So in the presence of IDH mutation, there is little signals of angiogenesis, and the host pericytes are stabilizing the vessels, and there is very little entrance of immune cells into the tumors, and this is more like a proneural phenotype. In the other side of the spectrum, in the presence of EGF receptor mutations, there is induction of SOX9, which is inducing all these expression of markers of pericytes. And these pericytes, these tumor-derived pericytes attached to endothelial cells, start expressing molecules typical for pericytes like PDGF receptor beta. So they make a more robust vasculature and they stabilize the blood-brain barrier. And the curious thing is this stabilized blood-brain barrier also allows the entrance of immune cells into the tumors. These tumors have a lot of proliferation and they are more similar to the classical phenotype. In the middle, in the absence of EGF receptor mutation, there is also induction of mesenchymal signals and angiogenesis, but these mesenchymal cells in the tumor do not attach to the vessels, so they don't stabilize the vasculature. There is a lot of leakage, and that gives rise to necrotic and hypoxia, and there is another way in which the tumors becomes highly aggressive, and this really resembles more the mesenchymal phenotype. We validated that, of course, in a cohort of samples from patients. You can see clearly that IDH mutations have a very few vessels compared, for example, with these very robust vasculature in EGF receptor mutations with highly dilated vessels. Something in the middle will be with the EGF receptor wild type or amplified. In the presence of the EGF receptor mutations, we see accumulation of phospho-EGF receptor that correlates with this accumulation of the vascular signature. We also measure the appearance of necrosis, which accumulates mostly in the absence of EGF receptor or the IDH mutations. And also the extravasation of IgG can be measured also in the human samples. We did some studies about the immune cells. It's not very profound, but we also noticed that the entrance of both lymphocytes, but especially myeloid cells, into the tumors in the presence of EGF receptor mutations. And what are the therapeutic implications of this? We know that Taxol and other derivatives can imitate the tau function stabilizing the microtubules. So we tested Epithelial D, which is a Taxol derivative that can enter the blood-brain barrier, that can cross into the brain. So we saw accumulation of acetylated tubulin and decrease in phospho-EGF receptor, and we were actually able to decrease the growth of the tumors in a model with EGF receptor amplification. Regarding the EGF receptor mutant, of course, it's given the tumors more aggressive features. They are growing more, but also they confer them sensitivity to some molecules. For example, ibrutinib, that I remember I mentioned you about, because it was described that BMX, which we have proven also that is upstream of, it is collaborating with EGF receptor in the activation of SOX9. So if we test ibrutinib in tumors with B3, there is a decrease in the growth of the tumors, and it doesn't have any effect in the absence of the mutation. And ibrutinib is blocking all the signal BMX, but also SOX9 stability and also the appearance of pericytes markers. And when the tumor cells attach to the endothelium, they express PDGF receptor beta, which is activated, and then become sensitive to sunitinib, for example, this molecule that blocks PDGF receptor beta, that also blocks the vasculature of these tumors, and also increases the hypoxia. So more or less, this is what we come out. We think that we could treat either tumors, proneural tumors, or tumors with a classical, with a mesenchymal phenotype. We could treat them with Taxol derivatives, And it's interesting because there is now a clinical trial going on with Taxol covalently bound to BSA, to albumin. It's called Abraxane. So I'm really expecting to see the results of this clinical trial. And in these other tumors, in the B3, in the tumors that have mutations of VHIR receptor, we think they're going to be especially sensitive to these molecules that target the tumor-derived parasites, like ibrutinib. And there is a clinical trial, I told you about it, in the Cleveland Clinic. Or also we can think of antibodies against CD248. And just to finish some models, I like this Waddington landscape. Probably you have worked in pluripotent stem cells, you have seen them. I like to see, imagine from progenitor cells in the brain. In the case of the present appearance of mutations in IDH1, or for example, amplification of PDGF receptor, these tumors start deriving into gliomas, but IDH mutant gliomas. When CDKN2, P16, P19 becomes amplified or mutated, these tumors become more aggressive. On the opposite side, by accumulations of more mutations in chromosome 7, EGF receptor amplification, of again P16, P19 amplifications, these tumors become more like a glioblastoma. And there is two different phenotypes depending on the presence of EGF receptor mutations, and probably P10 mutations have a similar role. So you have these three phenotypes, from the proneural one with a few vessels and few tumor cells, to the classical tumors with a lot of proliferation, a lot of tumor cells, a lot of macrophages infiltrating the tumor. And these are the phenotype in which the vessels are disrupted, there is a lot of hypoxia and very little tumor cells. And this is all controlled by the presence of IDH1, EGF receptor mutation, and the balance between all these signals, EGF receptor, angiogenesis, the tau MAPT. Now we are starting also to look around the tumor, and actually I don't have time to tell you a novel story we are working on, is that we are seeing that neurodegeneration that has been associated with the growth of gliomas is more common on mesenchymal tumors, and we believe is linked to this blood-brain barrier disruption, because we don't see it so much either in classical tumors or in proneural models. Remember, I told you what are the typical features of a glioblastoma, and we are now starting to focus our attention on this area of edema that is surrounding the tumor itself, because the surgeon is removing this contrast and has a tumor, this area here, but normally in 90% of the cases, the relapse of the tumors occur in this area of flare intensity, what we call the edema. And in this area, there is a lot of things going on. There is tumor cells, few tumor cells, but there is a lot of oligodendrocyte precursor cells. There is also neurons and microglia. And there is reactive astrocytes. And we believe, we believe that these cells are going to be important. They're going to change after surgery, and they are going to be important to understand how the tumors recur. And one thing that it was a little bit surprising for us is that the phenotype of the core of the tumor is also correlated with changes in this area surrounding the tumors. For example, the proneural tumors, even though they are less aggressive, they are highly invasive. You see a lot of GADPH, human GADPH surrounding the tumors that you don't see it in the mesenchyma. So contrary to other cancers, mesenchymal gliomas are not highly invasive. However, you see a lot of death of neurons surrounding mesenchymal tumors. And also another curious thing, that these are preliminary data, of course, we have seen a lot of reactive astrocytes surrounding the proneural tumor, not only close to the tumor, but it's even in the contralateral side. So we are starting to look at that. And I want to end up here by thanking the people of the lab, especially Ricardo and Berta. This is what I told you was most of the thesis of the PhD thesis of Berta, but it was a lot of work from both of them. Also from Pablo, Maria, Beatriz, Esther, Teresa, they all contributed to this work. And of course, the people from Hospital 12 de Octubre, particularly Juan Sepúlveda, Aurelio, Ana Ramos, Ángel, they contributed a lot to this work. And this is some of our funders. Here we collaborate with a lot of groups, and these are the collaborators, particularly in the stories I have told you today. And these are some of the collaborators in other stories that we are working on. So I'm gonna stop sharing.
Thank you, Pilar.
Thank you, Miguel. I hope it was well understood, because not seeing anybody is
yeah, no, it was a lot of things going on, but I mean, I think it's an excellent job, very clear, and I think we all enjoyed. So now it's time for questions. So everybody, I remind you, everybody wants to ask a question, please go to the preguntas part of the chat and ask there. In fact, we already have one that is from Carlos, from my group, that is very interested in the transdifferentiation process between the glioma cells and the pericytes, and how he's asking how well known is this process, and if this could be used as a targeted therapy, let's say.
Yes, I mean, in 2010, there were a couple of papers in Nature saying the transdifferentiation of glioma cell, this case, to endothelial cells. It was a big, I remember it was like a big hit. Everybody was talking about it. After that, for example, this transdifferentiation into endothelial cells hasn't come into nothing concrete. I don't see any treatment. It's not easy to see this transdifferentiation, okay? And then a few years later came this paper with Jeremy Rich and Shidenbao about this transdifferentiation to pericytes. I know sometimes there is some discussion, and I know these groups sometimes have been also argued that maybe it's not clear, but we have seen it very clear. We have seen, for example, B3, which is particularly expressed in the tumor cells. We see them in the pericytes, collateralization with the pericyte markers. So for us, it's very clear. It happens a lot, and it contributes a lot in aggressive glioblastoma. There's been some papers saying that it doesn't happen, but because they were using IDH mutations to identify tumor cells. And we know now that in the presence of IDH mutation, this process does not occur. So I think it's a process that happens in the presence of these mesenchymal features. And of course, I think it's going to be a very good target, for sure, for sure. I actually wrote to the people that are driving this clinical trial in Cleveland, because they were not particularly interested in EGF receptor mutations. And I hope after our paper, they're going to go at least look into it retrospectively, because I think it's going to be particularly interesting for tumors with these mutations.
Yeah, I think, well, I think so. Let's see what's going on with this trial. And I have also a couple of questions where people is trying to think about it, and I think about more questions. I have one that is, let's say, conceptual. I'm happy to see that you are really interested in understanding what is going on with the blood-brain barrier. And I think that you clearly demonstrated how these changes happen, but I'm not sure how these affect the functionality. I mean, let's see. It is clear, for the imaging techniques that the blood-brain barrier is not as functional as the normal one, because the dye enters into the tumor, and you can see. But then is the other part that you mentioned, that if tumor cells can also form a new type of blood-brain barrier, we will still have the same problem, that the drugs doesn't get into the tumor. So what do you think is the scenario? Do we have a blood-brain barrier that prevents the chemotherapeutics, or we can assume that no matter what, the therapeutics reach the brain, reach the tumor at least?
Yeah, that's a very good question, and I don't have a definite answer for that, okay? I saw recently some presentations from the group of Clinic Mayo. They're doing a very nice trial there, in which we are not only using drugs, but they're going to test if the drugs get into the tumor, and not only in the center, but in different areas of the tumor. They just started with this project, and I think it's terrific. I think only them can do it because it needs a lot of material. And they are starting to show some things that are a little bit surprising, that they were expecting, of course, in the vascularized rim, they were expecting more drug to be there, and they are expecting some things that they were funny. It's not just so simple. There are some mathematical modeling showing that if you get a lot of disruption of the blood-brain barrier, imagine it's like how you say manguera, I don't remember the word in English.
Manguera.
If you see this tube is highly disrupted, okay, so the drug can get out. The problem is you don't get the drug in all the areas of the tumor. So it's not just by breaking the barrier that you're going to be able to reach with the drug in all the areas of the tumor. So I'm not sure if the tumors with a lot of disruption of the blood-brain barrier are going to be easier targets. I mean, you know what I mean? I don't know. With this so disrupted vasculature, I don't think you get the drug into the tumors also easily. The same thing, you don't get the food for the tumor cells, you don't get the drugs.
Yeah.
So I think it's going to be a balance, and I don't have a definite answer. I would like to test different drugs, how they get into the brain, and really measure if they get into different regions of the brain.
Yeah, because at the end now, yeah, they are even still. No, you mentioned about the temozolomide, that is the only approved drug for this, and they keep using it, but not sure. I mean, if it's really reaching the brain, or the benefits that they can see come from another. From maybe the drug is altering the microenvironment as well. So I'm not sure.
Sure. I mean, it's supposed to induce the tumor cell death, but we are actually looking into that because there is actually many of when you test drugs for cancer, most of the assays you do them in vitro. You say, okay, is it toxic, and that's all, no? For example, with tau, it was the opposite. I remember Ricardo came into the lab and he said, no, tau, I don't know if it's doing anything, because when I put tau into the tumor cells, I don't see any difference at all. In vitro, nothing changes. Okay, I said, no, let's go into the mice. And then we saw this big difference. Or for example, we are collaborating with some companies testing different drugs. And we've been for a year or two testing this drug in vitro, which is inducing apoptosis, very nice results. And then we go into the mice and no apoptosis at all. And we see, again, changes in the vasculature. So probably it's because the dose that it gets into the brain is very low, and then the effects are very different.
Yeah.
And just with temozolomide, we suspect that it's not only a matter of cytotoxicity, but we are starting to think that it's also changes in the methylation pattern of the tumor cells, which are really controlling the phenotype.
Yeah.
And I think it's inducing some changes in these phenotypes between proneural and mesenchymal, but we are looking into that right now.
Okay. Then I was now good that you mentioned that the mesenchymal tumors are not per se more invasive than the others, as the contrary we could expect from the other tumors. But one thing that we have seen in our brain tumor models is the impact on survival is not only depending on the size of the tumor, but also in the migratory phenotype. So how is there any way that you can control this in your, I mean, we've seen the difference that you're clear, although small, obviously, difference in the survival of the mice. But have you seen this change of pattern, that maybe with these alterations, you have a more, let's say, round tumors encapsulated that do not go somewhere else, or do you have a more invasive tumors?
We haven't looked much into that. We have concentrated in the vasculature, but I remember some papers from OLIC2 describing OLIC2. And is the thing, OLIC2 is a typical marker of proneural progenitors, okay? So when OLIC2 is present in the gliomas cells behave a lot like progenitors, and they start migrating through the vessels and through the myelin sheets, okay? And these tumors are very, very invasive. And when they cut expression of OLIC2, these tumors become more round and concentrated. And it's true, it's funny, but it is like that. When you see them, again, if you test them in vitro, probably a mesenchymal glioma is more invasive than a proneural glioma, because proneural gliomas grow a very compact sphere, very typical like progenitors. And then mesenchymal tumors do not grow so compact. They grow very, they don't attach to each other. They attach a lot to the plastic. They are more invasive, like typical invasive assays. But then you put them into the brain, and it's very different. You can see the brain of a proneural glioma when we sacrifice the mice, and the tumor cells are all around the brain, and the mouse is still alive. Okay. But the mesenchymal is more compact. It's smaller if you want, because the size, it looks smaller, but then it kills the mouse more quickly. I don't think that's exactly the same in humans, okay? Because, for example, neurons in mice are very resistant to neurodegeneration. So maybe a proneural tumor is going to be as aggressive as a mesenchymal in the context of a brain, because now we are looking also in the neurodegeneration, and it's different in the different tumors, okay? So I think that the killing of a patient is a mix of the two things. Is the effect, the mass effect of the tumor and the edema that grows around, and also the invading tumor cells that are, of course, disrupting the function of the neurons in the brain. And I don't have a definite answer. I don't know which one is more aggressive.
Okay. So, and also another thing that with that experiment that you mentioned, that you showed with the stabilization of tau, no, with this drug, it's like it works well, no? So, what do you think that this very strong impact on the molecular markers, but not so great impact on the survival of the mice? So how? No, I mean it is also happening to us. No, you see clearly, no, with some SDAC inhibitors that you have a acetylation that you think that the tumor really reprograms itself, but at the end you manage to expand 1 week or 2 weeks maximum the life of the animal. So what we are missing, what do we need to do to improve, to cure, I mean, to get better results in the preclinics, but obviously this is also happening in the humans, that there is no way that we find a drug that expands the survival of the patients.
Yeah, it is true. It's true that we grow these models, these primary models, and the ones that grow are probably the more aggressive ones, are probably hard to treat. So we are really putting ourselves a difficult thing. Of course, we are setting this in a condition that is not exactly the same as in the patients, because we are just treating the animals sometimes from the beginning or sometimes from the middle of the growth. So we, for example, are going to try now to combine with surgical procedure in mice, which is not easy to do, okay, to see that maybe it improves. But it's completely right. I agree with you. It's a little bit disappointing that you can see only a small effect in the mice when you were seeing such effect in the change in the phenotype. The papers now that are looking into these different phenotypes of glioma, all of them are coming to this. You probably are going to have to combine. You're going to have to probably block the mesencymalization and then block the invasion, and you're going to have to probably block the two ways in which the tumor has to escape from the conditioning.
Okay, yeah, I agree. So I guess people is a little bit shy, no? And
maybe it was too much information. Anyway, the information is there, so you can go and take a look.
No, and I remember also to everybody that the session is recorded. So if you have registered to the session, you will have the chance to revisit the seminar, and also always Pilar will be available, I guess, by email to be reached and answer your questions if you mind. Okay, so Pilar, thanks a lot.
Thank
Hope
you
you
again.
can keep updating on the results on your research, and I hope to see you soon again, and with a clinical outcome.
I hope I can go there and not just like this.
No, I'm sure you will. I think also this project that you mentioned now to analyze this penumbra area will be very, very interesting. I think.
Yeah, that's a project we are starting actually with the neurosurgeons themselves, and we are going to be collecting samples from different regions. And actually, I mean, you're right, because you see other people are starting to do the same. And I saw some American groups are also looking into that, because it's true, the tumors are relapsing in this area, and we are concentrated on this area that is removed. And it's not the problem. The problem is what we left behind.
Yes.
And we are trying now to change our focus and look into this other area. I'm really excited with this other project.
Yeah, I take also the opportunity. I mean, it's not exactly my business, but the other day we were talking with the neurosurgeons, and they do have all these this new technique. Well, it's quite new, the microdialysis, where they can implant sensors in the penumbra area. They do for mostly for ischemic patients and not so for cancer, but they can measure the changes in the electrolytes and the functionality of the blood-brain barrier. And this, I think, this will also help to shed some light about the drugs, how we're reaching the drugs there, and the functionality.
That's interesting. That's interesting. I mean, we are trying to correlate our analysis of these different regions with the MRI imaging, because the MRI imaging, it only gives you the flare, but then there is a lot of other sequences like diffusion and perfusion images, and I think they're going to be informative also of what is there, and it's important to see it in advance. So if the surgeon has to take the decision if to go further, if to do a what they say supra surgery, or if they have to be very more strict to this contrast enhancing region. And also we are interested in something that for sure you're interested in, is the drug delivery, how to get the drug there. And we are going to try with the surgeons and with some biochemists and some engineering, with hydrogels or things to put the drug into the cavity of the surgery, or maybe delivering in another way to get there.
No, yeah, that's definitely a big field of interest, and also for us, obviously, especially in the pediatric population, where again the surgery has to be. I guess in adult patients is the same. You cannot really take a lot of tissue that is not tumor, no. But especially in a developing brain, so it's still even more important. So surgeons are very conservative, no, to not remove anything that is not extra. That is not really clearly a tumor, but the problem is, as happens with the adults, then you have relapse. Yeah. Okay, so we'll keep in touch. We will follow your research, and hopefully we'll have the opportunity to collaborate on this. And
maybe we can try your liposomes in our models.
Yes, we'll. This is not so easy also to reach the brain. We'll do our best. Yep.
Okay. Keep in touch, Miguel.
Thank you very much.
Thank you. Bye.
Bye-bye.